Answer will be pedology.
Sir, but in the answer key it is C, geology.
Geology. No, no.
Sir, that is why I was confused.
Wait. Let me check the answer sheet as well. I have the answer sheet because we always check the answer sheet as well. Just a minute. This was the in ecology part.
Sir, 133rd question. This was.
Just a minute. Just let me check the test number. Question belong to ecology. And question number 133. Perfect.
Yes, sir.
Wait. Question. So, Arpit, is there any confusion? Any other question as well?
Sir, the 131st question, sir. Sir,
Yes, this is the 131st. Okay. Any other question other than this one?
No, sir, that was 133rd, but 131st question is also a doubt.
What is the question? Send me, please, quick. Actually, I have to see the answer key, and I will recheck the answer key because we always recheck the answer key. But here the answer, what I told, that will be the answer only.
So that is why, sir, I also marked A, but
yeah, that is the answer. That is correct. That is correct. Next one. Sometimes what happened, but we always recheck that. Bulk of the nitrogen in the nitrate is fixed by symbiotic bacteria.
Yes.
Symbiotic bacteria will be the answer.
But lightning also fixed some.
Lightning 1% only. 99% from symbiotic bacteria, only 1% from the lightning.
Okay, sir.
Yes. Any more?
No, sir. Just
okay, fine. So I'll check with the answer key, that the question number 134, right? One question like that is quite possible when they sometime not, at least from out of 100 question, 2 or 3 answer always remains wrong in the book from where the question have been picked. Many times it happens. So we always, by the way, we always cross check with that. I go through all biology question answer sheet. Still I am making some answer sheet. So all biology answer sheet I make. I check myself. then only we supply to the student, right? I'll cross check with that answer key.
Okay, sir.
Okay, so we were here, WBC or leukocyte. So WBC or leukocyte we were doing, and in WBC, the peculiarity with the WBC that their main function is to protect body from the any kind of the external invasion or any kind of the external attack, right? So whenever the antigen attack to our body, these are the WBC which protect. So they do not have fixed shape. So their shape is amoeboid shape. So what is the benefit of that? Actually, normally the blood flow inside our blood vessels. But when the blood flow inside our vessels, what they do, they can come out of the blood vessels. While the RBC and platelet they cannot come out of the this one blood vessels. Why? Because they cannot change their shape. But blood vessels, there are certain creaks in the blood vessels. From there, they can leak out, they can come out and kill the germs. So even they can kill the germs out of the blood vessels, right? So that is the peculiarity of them. They phagocyte means they eat them and kill them, right? Normally their number is 6,000 to 8,000 millimeter cubic. In 1 ml blood, their number is 6,000 to 8,000. So I have already told you that this is very important question. I have seen this question twice or thrice. What is the ratio of WBC and RBC? So ratio of WBC and RBC is 1:600. In some book, it is written 1:60. That is wrong. The answer is 1:600. Right. That is the ratio of WBC and RBC in the blood. Right. Rise in the WBC is called leukocytosis. So leukocytosis is increase in the number of the WBC. Right. Abnormal rise in the WBC when they increases abnormally, right? And they start eating other cells. This is called leukemia. Leukemia is basically called blood cancer, right? So in blood cancer, these WBC they kill the other germs, right? Sorry, they start killing their own other than their other germs. They start killing their own cells, right? They start eating their own cells. That is called leukemia, means blood cancer. uh, 10 to 20 micrometer is the size of WBC. They have the nucleus, cell organelle are present. So, like the nucleus and cell organelle were absent in the RBC, but they are found over here, right? Formation of WBC, production of WBC basically take place in the bone marrow, right? And their maturity take place in certain organs. So WBC are actually, that is the important thing which we need to discuss. WBC are produced basically in our bone marrow, but for maturity, they go to the spleen, they go to the thymus gland, they go to the lymph node. So these are the areas where they go and they get mature. So formation of WBC takes place in bone marrow, but maturation takes place in thymus gland and our liver, thymus gland and spleen, right? Now, this is the type of WBC. You have to remember this chart. I have made this chart only for the exclusively for the medical examination. Look, so WBC normally they can be divided into two categories: agranulocyte and granulocyte. Agranulocytes are those WBC which do not have granules in their uh, protoplasm, right? So they are called agranulocyte. Normally, agranulocyte are of two types: lymphocyte and monocyte, right? These lymphocyte they may be. So further we divide lymphocyte, maybe B or T cells. So B lymphocyte and T lymphocyte. B lymphocyte are basically they are matured and they are formed in liver. B lymphocyte produce antibody. So that is the function of what is the antibody? Antibody is a type of protein which remains dissolved in our blood and it kills the other pathogen, right? In case if you want me to dictate any point and you want to note it down, you have to tell me, right? If you won't raise your hand, then I'll understand that I'll think that you know it already, right? So look, antibody is a type of protein which kill the antigen. Antigen means bacteria, virus, any foreign organism which can harm our body. So B cells, they produce antibody from their body, and that kills the pathogen. There are T cells, they are called thymus cells. they directly attack thymus cells. They are directly attack and they kill the bacteria and viruses. Further, granulocyte granulocyte are of two types: lymphocyte, monocyte. Right? Lymphocyte may be B lymphocyte or T lymphocyte. Right? They are non-motile or non-phagocytic. Right? So non-motile means they cannot move here and there, and they are non-phagocytic means they do not kill the bacteria. They do not eat the bacteria. They kill with the help of antibody. They produces antibody and destroy the microbe and toxic and tumor cells. That is important. The question which I have seen in AIMS, the question was that which of the following kill the tumor cell, and answer will be lymphocyte. You have to remember that our body have capacity to fight with the tumor cells, cancer cells. Tumor cells mean cancer cells. So our body have the capability to fight with the tumor or cancer cell, and they are the basically lymphocytes. They are 20 to 25% of the WBC. So their percentage is 20 to 25% of all WBC. Their lifespan from few days to years, right? Monocytes. Monocytes are the largest of WBC. They are the largest WBC. They are motile and phagocytic, right? They engulf the bacteria and cell debris, right? They eat the bacteria and some cell debris. Uh, their lifespan is 10 to 20 hours only. Now come to the leukocyte. So, which is the largest WBC? That is the question, right? Their lifespan, that is the question. Now come to the granulocyte, means those which have granules in their protoplasm. Their lifespan is 4 to 5 days, right? They are of three types: eosinophils, basophils, and neutrophils. Eosinophil, they increase in allergy, asthma, and hay fever. They help in dissolving the blood flow, right? That is the question. So eosinophil help in dissolving the blood clot in case the blood clotting takes due to internal injury. They are non-phagocytic. They are similar like lysosome. They destroy the protista. Basophils. Basophils release heparin. That is a question. Important question. Very important question. What is the function of heparin? Tell me, guys, what is the function of heparin? No one? Look. Heparin is a substance which is found inside our body. It remains dissolved with the blood. It do not allow blood to clot inside body. When due to injury, when blood come out of the body, heparin stop working and blood clotting start. Please note it down. Note down. Heparin is released from basophil. Heparin is released from basophil. Full stop. It remains dissolved with the blood. Full stop. It do not allow blood to clot. It do not allow blood to clot inside our body. Full stop. Plus, the next thing, histamine is released from here, right? Histamine is allergic, right? It is allergic response. When some allergen comes, then histamine is released and we feel itchy, right? They are least in count. That is not a question. That is not a question, right? Neutrophil. That question I've been asked. They are the most numerous of all WBC, the most abundant of all WBC. This question have been asked, right? So they are the most numerous of all WBC, right? Now, okay, they eat harmful germs, and that's why they are phagocytic, right? So you have to remember. So all things which is underlined or I have made the star, you have to remember all those, right? So because that are the potential questions. Any doubt in WBC?
No, sir.
Okay, great. So come to the next slide, next chapter. Blood platelets. Blood platelets or thrombocyte. They are basically responsible for blood clotting. That's why they are also known as thrombocytes. So normally you must have listened that when body get affected or liver is affected, then the number of thrombocytes decrease, right? So the normal in a normal person, the number of platelets are 1,50,000 to 3,50,000 per ml of blood. Increase in the platelet is called thrombocytosis, and decrease is called thrombocytopenia, right? They are colorless and they do not have nucleus, that they are non-nucleated. Lifespan of platelet may be 1 week. Formation of, that is important, formation of platelet takes place in megakaryocyte of vein, megakaryocyte of the bone marrow. Megakaryocyte are a larger size of a cell, and they are found in the bone marrow, and their exclusive their function is formation of platelet. So it was simple, and you have to remember from where the platelets are formed. Others are not potential questions. Come to the blood group. Blood group we have already discussed in the genetics part, right? Again, we are going to discuss over here. So blood group A, B, and O were discovered by Landsteiner in 1900. So this was a person, the name of the person was Landsteiner, who discovered the blood group A, B, and O. AB blood group was discovered by De Castillo and Stanley, right? There are basically four kind of the blood group: A, B, AB, and O. The name of the blood group is on the basis of antigen which is found on the surface of the blood group, right? So A blood group have A antigen but B antibody. B blood group have B antigen but A antibody. AB blood group have AB antigen but no antibody. That's why AB blood group is universal recipient, means it can take blood from the any other blood group. O blood group, it do not have antigen but have AB antibody. So this is the universal donor. It can give blood to anyone but can take only from the O blood group, right? So what is my question? Suppose that there is a person whose blood group is A. Try to understand. There is a person whose blood group is A, and he or she met with an accident. He or she is admitted to the hospital, and in place of A, the doctor transfused blood group B. What will happen with that person?
The blood cells will get destroyed.
Okay, Shania, the blood cells will get destroyed. Okay.
And it
Anyone
could
else?
be fatal.
Sorry, yes, very good. That can be fatal. Anyone else? Arpit? Any idea? Anna? Abhinav? Look, Sania have almost told correct answer, almost right. So what is the precise exact answer? What happens? Suppose that a person of blood group A is given with the suppose that a person his blood group is A, and he is given a B blood group, right? What will happen? The both of the RBC will get stuck. This is called clumping, or this reaction is also known as agglutinization. So clumping or agglutinization will take place, and due to which what will happen? Blood will clot and the person will die, right? That will be the death, right? This process, so you have to remember the clumping or agglutinization. This process called clumping or agglutinization, right? Now come to the, what is the antigen? That's all. That's what I also want to discuss with you. Can anybody tell me what is the antigen? How we can define antigen?
It's the harmful or foreign particles that enter the body.
True. Look,
It's
any
a foreign
harmful
body.
substance. Yes. Yes, Arpit, tell me.
I was saying that it's a foreign body.
Sorry.
I
That's
was
it.
saying that it's a foreign body that enters into the body.
Exactly. It's a foreign body that enter. And one more important thing which we need to add on. Whenever any foreign body which is not part of our body that enter into our body, it may be living or non-living, but when it enter, our immune system get triggered or respond against it, then this is the antigen. If immune system does not respond, this is not an antigen, right? Like you just for example, uh, uh, fill an injection with the water, pure water, and put this injection inside blood. So blood will enter into water will enter into blood. It's but the blood will not that the water will not the antibody formation will not take place again water, right? Where it can take place again any chemical, any other chemical, right? So but not for the water. So that is not antigen. So whenever a foreign body or foreign substance enter in our body, it may be living or non-living, and formation of formation of antibody starts again is called antigen. Antigen may be of two types. It may be living, means abiological or biological, right? So antigen may be living or non-living substance, both kinds of substance. So antigen can be just so antigen can be abiological, or biological. So abiological, it may be smoke, it may be dust, it may be chemical, or biological, it may be bacteria, it may be virus, it may be protozoa, it may be fungus, it may be pollen, anything, right? So look, when any antigen enter into our body, there are two mechanisms to fight with antigen. How the WBC fight with it? We have discussed WBC fight with it, but how does WBC fight with it? Suppose that bacteria have entered in our body. So there are two kind of the WBC. One kind of the WBC, they will release a protein, and this protein will, when this protein will come, this will remain dissolved with blood. When this bacteria will come in contact with this protein, it will kill bacteria. And this protein was antibody, right? So antibody was a type of protein secreted by WBC. These kind of the WBC, so that's how they kill the antigen. These kind of the WBC are called non-phagocytic, means non-eating. They are not eating any pathogen, right? Second kind of the WBC, they are called phagocytic WBC. These WBC eat the bacteria and die themselves, right? They work like suicide bomber. Now, what is the positive and negative blood? That we need to understand. Look, every RBC have a protein, right? RBC have a protein on its surface. This protein may be present over there, this may not be present. And if it is present, there is no any benefit. If it is absent, there is no any demerit. So this protein is called RH protein or RH factor. Basically, it was discussed in the rhesus monkey. That's why it is named as the RH protein or RH factor. If RH protein or RH factor is present on RBC, we will call it it will call it positive blood group. If RH factor is absent, if RH factor is absent, this is called negative blood group, and this is if RH factor is absent, this is called negative blood group, and uh, the RH protein will not be on the surface of RBC. So that's how the positive and negative blood group are formed, right? Discovered by Ladistiner and Weiner, and discovered on the rhesus monkey, right? Is that clear?
Yes, sir.
One very important thing: you can give positive blood group, you cannot give positive blood group. You cannot give a positive blood group to negative person. Why? If A positive, for example, if A positive blood group have been given to a person with A negative, right? You cannot give them. Why? If you will give them, then this RS protein will work as antigen, and this A negative blood group will start making antibody against it, and the same agglutination will take place. But you can give negative blood group to the positive one, right? You can give negative blood group to positive one. Why? Because negative blood group do not have RS protein, and uh, formation of uh, antibody will not take place in the blood group positive. Is that clear or not? Tell me.
Yes, sir.
Last thing I have told you. HDN disease. Who is going to explain me? I have explained this disease. I can show you. I have explained this disease in Mendelian genetics. Who will explain me HDN?
It's when the blood group of the mother and child is different. So during birth, there's a chance that the blood can infect, like blood can come through the. Oh my God. Basically, the mother needs to be injected with some
Very good. Yes, tell me.
other.
You are
The
close to the
mother,
answer. Yes. Now tell me.
the mother. It's because like the baby and the mother's blood group is different. So the baby's blood group can come into the mother through placenta or something
Very
else.
good.
So
Yes.
it's harmful for mother's health or life. So after giving birth, the mother gets injected with something.
Okay. All right. You are close to the answer. And let me tell you. Fine. So look, suppose that, try to understand. Suppose that mother is have negative blood group. Try to understand me. A mother have negative blood group and the baby have positive blood group. Normally, transfusion of blood do not take place between the mother and the baby through placenta. Normally, only nutrient and exchange of waste material and gases that take place through placenta. But sometime transfusion of blood takes place a little bit. Sometime means 99.9% won't take place, but 0.1% it will take place, and sometime it happens. So when transfusion of blood group take place, the mother's blood group is positive and baby's blood group is negative. So the mother body treat baby as antigen, and mother body start making antibodies against that baby. The first baby will survive and it will take birth with the jaundice. It will cause jaundice. But already antibodies have been prepared inside the mother's body. If the second baby is also positive, then what will happen? Second baby will die. Why? Because the mother body is already treating that baby as antigen, and that will die. This disease is called hemolytic disease of newborn, HDN. Is it clear or not? Is that clear to all of you? Tanya, got it?
Yes, sir.
Okay. Arpit, got it? HDN?
Yes, sir.
I have already discussed with those people who are there in the genetics part, right?
Sir,
Sana, clear?
Yes,
Abhinav?
sir.
Okay.
sir,
Right. Yes.
sir, this disease and hemophilia, sir, are they interrelated?
No, 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 no. Hemophilia is something different. That is genetical disease. It's not genetical.
Okay, sir.
Hemophilia is that X-linked recessive genetical disease. So this is hemolytic disease of newborn. So, but previously it used to happen. Second baby used to die if it have positive blood group. But nowadays, if doctor comes to know that the second baby is also, even the first baby is positive blood group, they give immunosuppressant to the injection to the mother and suppress the immune system of mother. Same thing they do with the second baby, and all the babies survive. That's what they do, right? So on that time when immunosuppressant is given to the mother, what happened? Immune system of the mothers remained very low and very inactive kind of immune system. On that time, mother have to take care of any infection. So this was HDN. HDN have been asked many times. HDN, but HDN is not a question of here human physiology. I am just telling you. This is a question of genetics. Blood group clear, guys?
Yes, sir.
Yes, sir.
Great. What is the lymph? So lymph is called middleman. It's a pale yellow color liquid which work as a middleman, and it also maintain the immunity. So look, this is the blood vessel and this is the tissue. So blood vessel do not directly exchange anything from the tissue. Blood cannot directly take anything directly from the tissue. It takes place through the lymph. Lymph remains filled between this yellow color substance called lymph. Lymph remains filled with the between the blood capillaries and tissue. If the cell have to take oxygen, it will take oxygen through the lymph. If cell have to take back the carbon, it have to take the carbon dioxide, it will take through lymph. If cell have to take the nutrient, it will take through lymph. If it have to excrete the nitrogen waste, it will excrete through the lymph. So lymph, that's why lymph is called middleman. What is the lymph? This basically the blood vessels of human, they are not completely transparent. So whenever blood flow through the body, so what happened? Whenever blood flow through the body, some amount of the blood plasma leak out of their wall. This leaked blood plasma remains filled with between the tissue and the blood vessel. This is called lymph, right? And lymphocyte cells, they also come over here. So lymphocyte cell and blood plasma means lymph. It means the complete lymph, and lymph is called middle, right? And there is, so every time if lymph will keep on filling down, if plasma will keep on leak down, so what will happen? First thing, that the amount of the blood will reduce, and the second thing, the plasma will keep on accumulating between the tissue and will get swelled. But that does not happen. Why? Because after a certain amount, there are a certain vessels called lymph vessels. These lymph vessels collect this. These lymph vessels collect this lymph and pour back again in the blood vessel. Lymph vessels have a node between them that is called lymph node, and they help in production of WBC. So lymph is made up of two things: lymph plasma and lymph capsule, that is lymphocyte, right? Lymphatic organs are called lymph node, tonsils, thymus gland is clean. Here the formation of lymph takes place. Tell me, is it clear or not? What is the lymph and what is the lymph node? Clear?
Yes, sir.
Okay. Very, very, very important part of this chapter. The most important part as per my view. This is the frequently questions are being asked. The first question comes from here: the blood clotting and blood coagulation. Both are the same. Blood clotting and blood coagulation are the same. Is it good or bad for our body?
It is good.
Good. Okay. Yes, Sania, good or bad?
Sir, can you repeat the question again?
So blood clotting or blood coagulation, right? So is it good or bad for our body?
Depends. Like, if it's like when it's injured, then it's good.
Very good. And when it is internal, it's bad. Right, correct. Absolutely correct. When blood clotting is taking place due to injury or outside of our body, that is good. because it do not allow extra blood, or it do not allow blood to go as a waste, right? So on that time, blood clotting is good. But when blood clotting takes place inside internally our body, that is bad, because that prevent the blood flow. So how does the blood clotting take place? The reactions of the blood clot that I have represented over here. Try to understand. You have not to cram that. You have to just remember this flow chart. When a person will fall down, the tissue will get injured and blood will come out and some of the platelet will disintegrate. So when injured tissue, they releases a type of chemical called thromboplastin. Thromboplastin, in presence of calcium and protein, it make prothrombinase enzyme. So thromboplastin make prothrombinase enzyme. In the same time, the blood platelets at the injured point, they get disintegrate and they release platelet factor 3. Platelet factor 3 is also known as platelet thromboplastin. Platelet thromboplastin in presence of calcium and protein again make prothrombinase. So finally, prothrombinase enzyme made by the both of the process from injured that is the injured tissues and from the blood platelet. This prothrombinase enzyme, it inactivate heparin. So what is the function of heparin? I told you a few minutes back. Heparin is anticoagulant. It do not allow blood to clot. So first, the function of heparin need to be stopped. It inactivate heparin, right? Then heparin inactivate and catalyzes the reaction. It convert prothrombinase enzyme, inactivate heparin. It convert prothrombin into thrombin. Thrombin is a solid protein, and then again this thrombin protein, it convert fibrinogen into fibrin. This is also solid protein. This thrombin and fibrin get deposit at the wounded part or injured part. They make a blood clot and do not allow further the flow of the fresh blood from the body and stop the loss of the blood. So that's how the reaction works. Is it clear or not? Tell me. And if you have any query, ask.
Clear, sir.
Okay. Now hemophilia, I have discussed in detail. Hemophilia is a disease where blood clotting do not takes place, and it's a genetical disease, right? This is also known as the royal disease or bleeder's disease. It's a genetical disease and sex-linked disease, right? We have discussed in detail in genetics part, so we need not to discuss over here. Samira, are you getting everything?
Yes, sir.
Heather, all well? Getting everything? Heather? Yes, Heather, are you listening me? Iram? Iram, why are you silent nowadays? Previously, you used to respond me. But nowadays, I don't get any response from your end. Yes, Iram? Is there any problem? Iram, are you listening me? Or is there any problem with your mic? Text me. So, blood circulatory system. Now it comes to the blood circulatory. Till that point, it was the blood. We were discussing blood, its property, its peculiarity, and all that. Now, blood circulatory system. So there is a two type of the blood circulatory system: open circulatory system and closed circulatory system. What is an open and what is a closed? In open circulatory system, look, first let me make you understand. So this is an insect, Arthropoda. Arthropoda is a phylum of insect, and they have the open circulatory system. Now, what is the open circulatory system? Look, this is the insect, and inside there is a blood remains filled like this way. An organ bath on the blood. Organ just bath on blood, right? Organ keep on bathing on the blood, and the space where the blood remains filled is called lacuna, right? And What happened? Suppose that even the roundworm, starfish, insect, they have the open circulatory system. So if they get the injury, complete blood come out, and they exchange the oxygen and carbon dioxide directly from the skin. So there is no artery or veins in open circulatory system. Blood remains filled in spaces called lacuna. The organ bath in the blood may or may not have heart. Heart may be present or may not be present. Right? Now closed circulatory system. In closed circulatory system, blood and O2 that comes out, right? So blood and O2, so blood plus O2 come out of the heart, and it is supplied to the whole tissues through a particular pipeline called artery. So artery supply O2 or oxygenated blood to the organ. Organ burn the fruit and convert them into carbon dioxide. The carbon dioxide is released and it reaches to the heart with the help of veins. So blood flow in these closed vessels. That's why this is called closed circulatory system. So blood flow in closed vessels is written over here and is found in the mammal. Blood flow in closed vessels. All vertebrate, means those which have the backbone, they have closed circulatory system, and their heart is divided into chambers, right? The Pisces or fishes have two chamber heart. One is filled with the pure blood, one is impure blood. Amphibians have three chamber heart, pure, impure, and mixed blood. Reptile have three chamber heart, pure, impure, and next, that is the mixed one. But crocodile have four chamber. That is exception. Apes have four chamber, means birds have four chamber. Mammals have four chamber. Whale is a mammal, that's why it have four chamber. Bat is a mammal, that's why it have four. Right. So this was the type of circulatory system and the chamber. So it was simple. There was nothing difficult. Now, what is the double circulatory system? Even this is this you need to understand just for sake of your knowledge. This is double circulatory system. The blood make two cycle of the heart. That's why this is called double circulatory. Look, blood coming out of the left auricle, it goes to the lungs and the impure blood, right? So impure, don't start from here. Let me start from. Look, this is the tissues. In tissues, we know that burning of food keep on going in our mitochondria, and due to burning of the food, carbon dioxide is produced. So blood take carbon dioxide. We have discussed all things, right? All those things. Blood take the carbon dioxide, and that becomes deoxygenated blood. So it is taken by the tissues, and the right this blood, this impure blood, it reaches to the right auricle, right, or right atrium. Right auricle or right atrium, it reaches to the right atrium. And when it reaches to the right atrium, what happens? From right atrium, impure blood, and which takes the arteries, veins take it to the right atrium. And the vein, this is the largest vein, this is called known as vena cava, right? So pre-vena cava and post-vena cava. So they are just vena cava are a type of largest vein which pour blood into right atrium. From right atrium, blood come to the right ventricle. From right ventricle, this blood, deoxygenated blood or impure blood, goes for purification. Means the blood with CO2 that goes to the lungs, right? And the pipeline which take impure blood from the heart to the lungs is called pulmonary artery. While the function of artery is to carry oxygenated or pure blood. Pulmonary artery is the only artery that carry the deoxygenated or impure. So blood reaches to the lungs and then carbon dioxide get diffused. Now what happened? Blood get dissolved with the oxygen, right? Pulmonary vein is the only vein that carry deoxygenated blood. Pulmonary vein take this oxygenated blood to the left atrium. From left atrium, it goes to the left ventricle. From left ventricle, it goes to the aorta, and aorta spread it to the whole body. So in the nutshell, there are two cycles of the blood circulation. First cycle, when blood goes from the heart to the lungs and come back to the heart. This cycle is called pulmonary cycle. Pulmonus is a term. Pulmonus means lungs, and pulmonary means to the lung, right? Then blood flows between the tissue and the heart. This is called systemic circulation. Systema means tissue, right? So this is called systemic circulation. Okay, so there are two kinds of the circulation: pulmonary and systemic circulation. That's why we call human blood circulation is a double circulatory system. Tell me, did you got that double circulatory system or not?
Yes, sir.
Okay. Simple. I have made this diagram, so you need to understand just a few things. First thing which you need to note down the weight of the heart. So in males, heart is larger, and female, the heart is smaller. The male, the size of heart is 280 to 340 grams, but in female, this is 230 to 280 grams. So in female, the size of the heart is smaller. Heart remains surrounded by a membrane that is called pericardial membrane, and around this membrane, a pericardial fluid remains filled that protect heart from external injury. Look, this is the heart. This is right atrium. This is left atrium, and this is superior vena cava. So impure blood from the upper side, upper body organ, come from the superior vena cava. Then there is an inferior vena cava. Impure blood from the lower body organ come to the inferior vena cava. It reaches to right atrium. From right atrium, it reaches to right ventricle. From right ventricle, it goes to the this pipeline, left and right pulmonary artery, and reaches to the lungs. Again, the pure blood come back to the left atrium through the left pulmonary artery and left pulmonary artery. Right. Sorry, left pulmonary vein. It comes through the left pulmonary vein. Come to the left atrium, then it goes to the left ventricle. From left ventricle, this is called aortic arc. It goes to the aortic arc. This is called subclavian artery. It takes blood to the clavicle part, this shoulder part, clavicle part. Then this is common carotid artery. It takes blood to the brain, common carotid artery. Again, left common carotid artery, it takes blood to the left side. Then subclavian artery, again, this part. Then come back lower side, this is called aortic arc, right? And from the back side of the heart, it goes down and descending aorta, it supplies the blood to the whole body. The thing which you need to remember, very, very important, who supply blood to the heart? Can you tell me? Tell me the artery which supply blood to the heart, because heart itself require oxygen for the working, because they keep on working 24/7. Tell me. Tell me, guys. Which supply? Tell me, guys. Which supply blood to the heart? Nobody's. Come on, guys. This question have been asked. Look. Right carotid artery. Note it down. Carotid artery. Sorry, coronary artery. I'm sorry. Right coronary artery and left coronary artery. Note it down. Coronary artery supply oxygenated blood. Please note it down. Coronary artery supply oxygenated blood to the heart. Let me write it down. Coronary artery supply oxygenated blood to heart, and coronary vein supply deoxygenated blood from heart, right? So that is the coronary artery and coronary vein. So coronary artery bring oxygenated blood to the heart and supply to the heart, and coronary vein take that, right? That you need to know. So it was simple. There was nothing much more interesting and much more important. Come to here. Here, what you have to remember from this diagram, you have to remember only the types of valve which are formed and what is the function of those valves. That you need to remember the valve. You may get question from these valves, right? So this diagram is important. It is lateral section of the heart, right? I will tell you the number of the all these valve, and you have to remember only those valve which just by those valve which I just demarcate, right? So first, try to understand. From lower body organ, deoxygenated blood goes to the right auricle. RA, that is right auricle. The backflow of blood is prevented by Eustachian valve. I have just highlighted it. The backflow of the blood is prevented by Eustachian valve, right? So Eustachian valve is a valve which prevent the backflow of the blood, right? So first you remember Eustachian valve. Then the upper or superior coronary, superior vena cava, it do not have any valve, right? Then the coronary vein, try to understand. Coronary vein bring deoxygenated blood from heart itself and drop into right atrium. The coronary vein in backflow in the coronary vein is prevented by Thebesian valve. This is called Thebesian valve. Either you call it Thebesian valve or you can call it coronary valve. So Thebesian valve or coronary valve, it prevents the backflow of the blood into coronary vein, right? Then blood come from the right atrium to right vein. There are some valve which are known as the tricuspid valve. These tricuspid valve do not allow backflow of the blood. These tricuspid valve do not allow the backflow of the blood into a right atrium, right? Tricuspid, there are three half cup shaped valves, that's why called tricuspid valve, right? Then next one, the blood goes from the right ventricle to the pulmonary artery, means for the lungs. It is guarded by pulmonary semilunar valve, right? Guarded by pulmonary semilunar. It do not allow black. Again, the pure blood come to the right atrium from left pulmonary vein. It reaches to the left atrium. From left atrium, it goes to the left ventricle. It is prevented by, means backflow is prevented by mitral valve. It is prevented by mitral valve. That is also known as the bicuspid valve. Bicuspid or mitral, right? Then blood goes to the aorta. aorta and supplied to the rest of the body. Backflow of blood from the aorta is prevented by aortic semilunar valve. These are the basically 6 valve. You have to remember the name of all these 6 valve, right? So, and tell me if you have any doubt regarding any of the valve. Guys, go through with it. You have to remember the name of all 6 valve, where they prevent and what they prevent. Where they prevent backflow and what they prevent. Go through with it and tell me, is it clear or not? Done? Sania?
Yes, sir.
Samara, is it clear?
Yes, sir.
Hana, clear? Abhinav?
Yes, sir.
Iram? Fine. So remember the name of the valve. Heartbeat. So it's simple. Heartbeat originates from a point that is, this is situated in the same thing, this direction. This is called sinoatrial node, SA node. So SA node is also known as the pacemaker, right? So heartbeat originate at the pacemaker, right? That 72 per minute heartbeat is maintained by the pacemaker. That is called SO node. From SO node, current flow to the left atrium, and the current mainly come to the AV node, arioventricular node. So SA node is found in the right upper side or back side of the right atrium. Then between the right and the left atrium, there is a node that called arioventricular node, AV node, right? AV node. So from AV node, it brings the current to the bundle of His, right? To the bundle of His, right? And here from here, this is transported, or it remains spread across this current. So bundle of His bring this current between the right and left atrium, this muscular part, and then it is spread across the body with the help of fiber called Purkinje fiber, right? So you have to remember the pacemaker is called SA node, and where is the OV node situated, and what is the function of Purkinje fiber. And heart, the function of heart is controlled by the medulla oblongata. That is the hide brain part, right? Medulla oblongata or hide brain, right? So it might be clear to you. It's simple. Okay. Heart sound. First heart sound comes from the partially closure of bicuspid and tricuspid bone. Okay. And partially by contraction of muscles of ventricle, and that sound is the lub. So lub is first sound. It comes from closure of bicuspid and tricuspid valve and contraction partially from the contraction of ventricle. The second sound is dub. It comes due to the closure of the semilunar valve, right? And heart murmur, when this sound comes lubs as like this way, then this is called heart murmur, and this is a type of disease. It is a disease sound. So basically, the doctor listen with the help of a stethoscope. Listen, this murmur sound is there or not, right? So if murmur sound is there, then there is some heart disease. ECG, very important question. I'll give you this question in your test as well. So try to understand. All the waves have been asked. PQR complex. The most frequently asked question is what does mean by QRS complex? What is P wave? What is T wave? The question will come through the diagram only. And multiple times the question have been asked. So you must have seen this diagram in the heart cardio hospital, right? And there is a machine which measures this diagram. So what is this diagram? This diagram is a graph of the pulsation of our heart. The machine which calculate this diagram or this pattern, that is called electrocardiogram, and the machine and the graph which we get, that is called electrocardiograph. Electrocardiograph is made on the basis of pulsation of the heart, right? So many of you have seen this machine. So it makes three kind of the wave: P wave, QRS wave or QRS complex, or T wave. Now what does mean by these waves? P wave is called atrial depolarization. This is atrial systole begins. This is when atrial systole begins, right? Atrial systole begins means contraction of atrium, right? That forces blood come to the ventricle. So blood come from auricle to ventricle, right? This is called atrial contraction, right? Atrial get contract and blood come to the ventricle. So due to contraction of atria, the formation of P wave takes place. And contraction of atria is also known as the atrial depolarization. Then what does mean by the QRS complex? Try to understand. From here to here, this is the QRS complex. When now the blood come to the ventricle, when blood get filled in the ventricle and ventricle contraction, when ventricle get contract, these both wall will get pulled, right? So when ventricle will contract and AV wall will close, formation of QRS complex will take, right? And due to contraction of, again, due to contraction of ventricle, that is ventricular contraction, the semilunar wall will open and blood will go out, right? And after that what will happen? Again the semilunar wall close and blood poured into atria. Blood flow will start into atria. That due to this, the formation of T wave will take place. This is called ventricular repolarization, right? When ventricle will swell and the blood will start coming, means get relaxed, and the blood will start coming into atrium. This is called ventricular depolarization. So this is the meaning of these waves. Go through with this chart, this diagram, and ask me if you have any doubt. If you didn't got it, ask me. You will get question from here. If you didn't got it, then you will miss the question from here. So don't take it for granted.
You can explain again the QRS.
Yeah, QRS complex means when now the blood is filled in the ventricle. When blood will fill with the ventricle, this bond will tricuspid and bicuspid wall will close, right? Now the ventricle will contract, get compressed, right? Ventricle will contract and pushes AV this wall to close. This makes the QRS complex, and this process is called ventricular depolarization.
Okay.
Disorder of heart, hypertension, high blood pressure. When the blood pressure become 140 to 90, that is called high blood pressure or hypertension, right? Yes. Okay, so what is the CAD? This is also very frequently asked. CAD and MGR. These are the two most frequently asked disorder. So what is the CAD? CAD called coronary artery disease, or the same disease is also known as CAD. In short, is called CAD, or it is also known as atherosclerosis. Atherosclerosis, right? So these all are the same thing: atherosclerosis or coronary artery disease, right? So right, CAD or atherosclerosis. So what happened in atherosclerosis or CAD? For deposition of fatty substance takes place. You must have listened this thing. It's very common. So deposition of fatty substance takes place in the some of the our muscles, right? Heart muscles. Deposition of fatty substance. Fatty substance means that is cholesterol. Takes place in the muscles of arteries. In arteries, right? And this deposition is called atherosclerotic plaque. This deposition, and that causes blockage, right? Now, in common language, that is called blockage of artery, right? And it caused due to LDL, low density lipid. This is called atherosclerosis plaque, or this is called the CAD or coronary artery disease. Is that clear or not? Tell me.
Clear, sir.
Okay, then there is a angina. Uh, sorry, angina. Angina is also known as angina pectoris. I have written over here. This is also known as the angina pectoris. In angina pectoris, the term actually the pain start from here. Pain start from here and it travel to the chest and come to the chest, right? And this disease, when it take place, when enough enough oxygen does not reach to the heart. Does not reach to the heart, right? And the person uh, person usually experiences the chest pain. Chest pain is experienced. That is called angina. Angina, the chest pain, that is called angina, right? And this is common. The most important thing, common among the young and middle-aged people. You and me, middle-aged and young people. That is called angina, right? And heart failure, that is a heart means heart do not supply that amount of the uh, oxygen or blood which is required. That is called heart failure. Heart failure doesn't mean that heart stopped working, right? That is called heart attack. So both are the different things. Okay. Heart failure does not pump blood effectively, and heart attack means the heart stop working. Is that clear? All disease. CAD is very important. Angina is very important. Frequently asked disease.
Yes, sir.
So yes, go through with your NCRT and go through with this chapter, and you have just 2 minutes, and let me know if you have any doubt in this chapter. Then I will move to next chapter.
Sir, can you repeat the topic from this chapter from which like questions
Sorry,
are mostly asked?
which one?
Can you repeat the topics from which the
Yes,
questions are
yes.
mostly asked?
So first, this one, CAD and angina in this disease part. The most important part, this one, from this diagram, from this diagram, this graph, right? Then these valve, right? So first important thing, that diagram, that graph part, this diagram. Then it is not that much important. This is also not important that much. This not this one. Very important. So first important point, that ECG diagram. Second important, this blood clotting and blood coagulation part. And then third, those diagram, valve. And the fourth very important part, this one, WBC, types of WBC. These are the four important. From where there is a 99% chances question, if the question will come from the circulatory system, definitely question will be from these four topics. Clear?
Yes, sir.
Okay. Any more doubt?
No, sir.
No. Okay. So then next chapter is, that is the excretion part, right? Excretory system. So let's move into excretory system. Start with that. Excretion part. Small chapter. So this is just for your understanding why the excretion takes place, why we need to go for urine, why we require to go urine, or why we require to eliminate urea from our body. Why it is important. So first, what is excretion? So removal of waste is not excretion. Removal of waste food is called defecation. There is a particular term. If somebody is using excretion term, that is a wrong notion. So I have written over here, the removal of undigested food is called defecation, not excretion. Then what is excretion? Excretion is just a removal of nitrogenous waste from our body is called excretion. So basically the excretory organ are skin, from where the sweat come out, sweat gland, then urine and all. Excretion. So why we need to go for urine? We eat three important things, which is part of our nutrition and part of our body, nutritional food. So carbohydrate, fat, and protein. Carbohydrate are just sugar, and extra carbohydrate, which is not required by body, it is stored in our liver and muscles in the form of glycogen. So there is arrangement of storing extra sugar. We eat fat, and whenever we eat extra fat, that the extra amount of the fat get deposited in a special kind of the tissue, adipose tissue. We have discussed many times the adipose tissue. They are found in our cheeks, they are found in our stomach, tummy part. And the protein is the most required part of our body, because why? Because our whole body is made up of protein. So every day the formation of new cells are required. So protein is required in higher. So protein is made up of amino acid. Extra protein, the protein which is required by our body, that is utilized, but extra protein cannot be stored. There is no storage of extra protein. So extra amino acid, what happens with them? Body do not throw anything as a waste, right? So what happened with the extra protein? Proteins are converted into amino acid. This is the formula of amino acid, RCOOH. What is this R? This is the alkyl group. Alkyl group. This COONH2 amide group, it is removed from here, and alkyl group is converted into sugar, so stored in the form of sugar. Now this amino group is converted into urea. The formula of urea is NH2CONH2. In the liver, right? This process goes on in liver. What happened in the liver? This is called urea cycle. In urea cycle, what happened? In urea cycle, this amino acid goes and alkyl group is converted into sugar, and this amide group is broken down and converted into urea, and is removed from the body with the help of urine, right? So we eat extra amino acid, that's why we require to go for urine. Is that clear?
Yes, sir.
Okay. Now mode of excretion, very important. You have to remember the example of all these above. Look, we go urine, and we, with the help of urine, we remove the urea. But every organism do not remove the urea, or every organism do not go for urination. Then what happens? There are some organisms which excrete ammonia as a nitrogenous waste from their body, and those organisms are called ammonotelic. Example, they are found in the excess of water like earthworm, leech, and arthropod. Means they are ammonotelic. But those which are found in excess of water. Then come to those organisms which remove urea as nitrogenous waste from their body. They are called ureotelic. They are found in moderate water. So when earthworm is present in high amount of the water, it is ammonotelic. When it is found in moderate water, it is ureotelic. And then frog, turtle, and human, etc. There are some organisms which remove uric acid from their body. They are called uricotelic. They remove uric acid from the body.